Okay, Biology 107 fans, we're going to talk about nucleotides and the genetic code. I'm going to split this into two flip lectures so that they're a little smaller and bite-sized if you'd like. Let's get started right away. We finished talking about proteins, and if we think about this in terms of what was the first self-replicating molecule that came into being on the earth, and proteins are not it. And proteins are not it because, number one, they cannot, right? There is no self-replication, and they do not store, so no self-replication, and they do not store long-term information, okay? And those two things prohibit protein from being that first self-replicating molecule that had, excuse me, that had material in it. Excuse me again. Okay, I'm sorry about that. The phone ringing, everything happening. Proteins were not the first self-replicating molecule that we know about, and so today we're going to talk about nucleotides and RNA and DNA. We've already talked a little bit about the structure of RNA and DNA, and the structure is sugar. All right, let me get a pen that actually is useful. All right, so every nucleotide is going to have a sugar, a base, and a phosphate group. And you should, at this point, be able to draw some of that and have it correct. The primary sequence of DNA, when we're talking about a string of DNA, is the sequence of nucleotides. And we usually, sometimes we'll draw DNA that looks like this. It's a line from the 5' to the 3' end. And what that means is that this is a string of one nucleotide bound to another nucleotide, bound to another nucleotide, bound to another nucleotide. Sometimes you'll see this string as a string of letters, A, C, G, A, C, T, A, G, etc. Okay, this once again is showing you the links between nucleotides. And we call this the sugar phosphate backbone based on the structure that we're talking about. So in the next image we'll see what I'm actually talking about, but there are rules, just like there were rules for proteins. You always draw a protein, so proteins are always drawn from the N to the C terminus. The N is on the left, the C terminus is on the right. You always draw DNA from the 5' end to its 3' end, and I'll explain what those are right now. In this image on the right showing a nucleotide, you see a base, right? It's a nitrogenous base. It has a nitrogen in it. We have a 5-carbon sugar right here. That is our ribose molecule, and depending on what's on the 2' position, that'll determine if it's DNA or RNA. And we have a phosphate group. And the linkages between these, right, are what create a nucleotide. These are all covalently linked together. The base is linked to the 1' carbon, the phosphate is linked to the 5' carbon. You should know that, right, deoxyribose is what we have in DNA, deoxyribonucleic acid, and for RNA we use the hydroxyl on the 2' position, and that is very important. When we link together two nucleotides, so here is our first nucleotide that we're going to link to this second nucleotide, what happens is a reaction called a condensation reaction, and we've seen a condensation reaction before in the formation of a peptide bond. So what comes out when you have a condensation reaction? You get the water is a product of this reaction. So when you add one nucleotide to another, you get a link between the two nucleotides, and one water molecule comes out. So here's the first nucleotide. Alright, here's the first nucleotide. You see the base, the sugar, the phosphate, and on the 3' position you have a hydroxyl group. Hydroxyl groups, very important in biology, they're very reactive, meaning they like to make bonds. Alright? And so this hydroxyl group can interact with the phosphate on the next nucleotide here, and what happens is that you end up getting a link between this 3' end to the 5' end of the next nucleotide. Now, what's interesting about this image, it came from a book and it's wrong, because you need some energy to make a covalent bond. And so let me add, what it means is we're going to have, okay, let's just quickly draw, right, here's a base, and if this is DNA, so we have this hydroxyl group, you have CH2, OPO, okay, there's a nucleotide, and what actually happens is you have the second nucleotide is going to be a molecule that has 3 phosphates, a triphosphate. So you have this, you have the base, but instead of just having one phosphate on the end, you actually have, this is poor planning, so you have 3 phosphates, 1, 2, 3. Okay, they all have to have their correct bond, so each one is going to have a double bond to an O, and it's also going to have a single bond to an oxygen on the bottom. And what this does is the terminal 2 phosphates here, these provide energy to make the bond between the first phosphate and this hydroxyl group. Okay, so what ends up happening is that 2 phosphates will come off, PPI comes out of this reaction, and you end up with, I know this isn't beautiful, but you end up with this particular linkage, O, P, O to that next CH2. That's my 5' carbon base, right? Alright, and then if I finish drawing this one up here, right, this one only had 1 phosphate. Okay, and this OPO here is a phosphodiester bond. And ester has to do with having an oxygen linked between something and then another oxygen, and this is a diester. So you have an oxygen with a phosphate linked to an oxygen. A phosphodiester bond. So this condensation reaction results in water, the loss of inorganic pyrophosphate, it's PIs, that's what it's called, and the energy that was in the bond here is actually transferred to the bonds of the phosphodiester bond. So the primary structure of DNA or RNA is just the string of nucleotides. And we draw them in multiple ways. I already told you, you can draw them with a 5' to a 3', and we're talking about the line is actually nucleotides. Or sometimes you see it and we just use the bases, right? A, G, G, C, C, etc. And you always do 5' to 3'. But there's also some secondary structure associated with strings of nucleotides. And you all know what this is. This is based on the double helix. And what that is you have, you know, if you look at DNA, you draw it up and down, right? So if you have DNA and it goes 5' to 3', and then you always have a second strand of DNA right next to it. DNA does not like to be single-stranded, it's double-stranded. And, right, we have, it's funny because a lot of people draw it with, it looks like a ladder. And those are non-covalent bonds in there. So the rungs on the ladder are non-covalent bonding. And what actually happens is the ladder twists. And that creates the helix. You should know that A pairs with T in hydrogen bonding, and C pairs with G. Now in RNA, that means that A pairs with U, and C still pairs with G. So the secondary structure of DNA is due to the two strands, right? Here's one strand, and here's the other strand, and they line up anti-parallel. Right? Parallel means going the same distance between them the entire way, but one is going in one direction and one is going the other direction. That's why it is anti-parallel. The hydrogen bonding is shown between the nucleotides, between the bases, and the sugar phosphate backbone is because there is a sugar. Okay, here's a sugar, phosphate, sugar, phosphate, sugar, phosphate. So the whole structure of DNA is, the structure is based on the sugar phosphate backbone. Okay? This is once again showing you now, if you actually take that sugar phosphate backbone of the anti-parallel strands and you twist it, you get this double helix structure. It's like a winding staircase with the bases facing the inside, and you have the sugar phosphate backbone shown in red, right? So the sugar phosphate backbone shown in red, and in between that the bases are inside here, and they're hydrogen bonding to one another. Okay, if you study DNA, you're going to figure out that there are these major and minor grooves. I don't care so much about that. I do care that you know that DNA is highly negatively charged, and it's because of the phosphates on the outside. Remember, each phosphate has a negative charge on it, and this makes DNA one of the most negatively charged molecules in your body. The secondary structure of DNA was first identified by a couple of people. Watson and Crick are very famous for this. And come in. The secondary structure of DNA was originally identified by Watson and Crick, and they had to use the data that was given to them, that was generated by Rosalind Franklin. And actually this has become very important in the history of science because Rosalind Franklin died very young, and she was not able to receive the Nobel Prize for this, whereas both Watson and Crick did receive the Nobel Prize. And it turned out that Watson and Crick couldn't have figured it out unless they had the data. This is the actual data that was produced by Rosalind Franklin. And it was her supervisor, so it was Rosalind Franklin's principal investigator, the person she worked for, that gave Watson and Crick the data they needed to determine the secondary structure. And what it demonstrated, what they figured out, was that the DNA was wound into this double helix, and this was based on the distance between each of the pairs of nucleotides as they complementary base paired between the two sugar phosphate backbones. What was that? What did I just... So the two sugar phosphate backbones, right? If I draw one DNA strand like this, 5' to 3', and here are the bases that are sticking off of that. So A, C, T, C, G, G, A. The second strand is going anti-parallel, and so let's draw 5' to 3' in that direction. In this case, we're going to draw 5' to 3' in that direction. And there are nucleotides on this strand that complementary base pair with the other strand, and this is by hydrogen bonding. Notice I put two dotted lines. The bond between A and T, okay, A pairs with T, and it does it with two hydrogen bonds, and C pairs with G, and it actually has 3 hydrogen bonds. So 3 hydrogen bonds. Alright, and so this would be a C, G, A, G, T. Okay, 2, 3, 2, 3, 3, 3. Okay, and actually the latter, they should be the same distance, but you get the idea from this. You have anti-parallel strands of DNA that are hydrogen bonded between them that generates the double helix structure. And Watson and Crick discovered this with the help of data from Rosalind Franklin. And it kind of sucks because Rosalind Franklin should just get just as much reward from this, but you cannot receive the Nobel Prize posthumously, which means after you're dead, and since she died young, she was unable to receive it. Alright, so now we're to the question, can DNA be the original self-replicating molecule? And what do we say is good about, being a living being? Well, we know that you need to have a dependable source of information. We need that blueprint. And it turns out DNA is a very, very, very stable, dependable source of information. I drove across the country with DNA in my car, and the DNA is just fine. It doesn't degrade. It's very strong, and it can stay for long periods of time out at regular temperatures. Now, interestingly, it doesn't fulfill the requirement for being able to self-replicate, and that's because it isn't catalytic. Alright, so this is what knocks it out of the running for being the first self-replicating molecule. It can't do a catalysis reaction. It can't act as an enzyme, and therefore it actually can't help itself replicate. So DNA cannot replicate by itself. So it has one of the features required, but it doesn't have really the second major feature. So the two major features are being able to self-replicate and having the information stored in it that would allow for heredity and evolution to occur. So DNA is out. RNA. What about RNA? Well, DNA and RNA structures are very similar, as we've already seen. Okay, this is showing you the sugar that has the deoxyribose, and this is the sugar that you find on DNA. And here you see the ribose molecule, and the big difference is this hydroxyl group on the 2' carbon. Everything else is exactly the same. Here is where instead of a hydroxyl we would have our base, and instead of this hydroxyl we would actually add our phosphate group out here, and that's now a ribonucleotide. Same thing here, right? So if we had a phosphate group here and a base here, this would be a deoxyribonucleotide. I think you get the picture. And this one is a ribonucleotide. But they're very similar, and the only major difference is this hydroxyl group on the 2' position. Well, so the primary structure of an RNA molecule is, right, it's going to be a nucleotide, 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 nucleotide. What am I talking about? I'm talking about the sugar phosphate, sugar phosphate, sugar phosphate backbone that we talked about previously. And that's the primary structure, is just a string of nucleotides. But there's actually a secondary structure, and it's due to complementary base pairing. You can see this complementary base pairing is huge in nucleotides. And what it means is, even though you have a string, here they're showing you in yellow, right, is the yellow is the sugar phosphate backbone. And each of the little blue and green tabs is a different base. And so you see that you get G's pair with C, U's pair with A, so U's pair with A in RNA, and G's pair with C, right? So C's pair with G, and A's pair with U. And that leads to these particular structures called stem loop structures. RNA is filled with stem loop structures, and they form due to complementary base pairing. You might be asking, well, why doesn't DNA do the same thing? It has the same idea. Well, you know the answer to that. DNA already is complementary base paired already because it has the double helix structure, right? So you get complementary base pairing because of the structure already. The double helix is secondary structure. Secondary structure for DNA, double helix. Secondary structure for RNA are these loops. Stem loops. Okay? DNA, right? We don't really talk about tertiary structure, but we can see tertiary structure in RNA pretty easily. This picture is showing you a RNA strand. Okay, so I'm going to go along, and this is the RNA. These are made up of nucleotides. This is the sugar phosphate backbone. And this whole thing is just one strand of RNA. You start at the 5' end, you end at the 3' end. This is linked. How are they all linked? They are linked through phosphodiester bonds. Those are covalent bonds. But then you also have secondary structure, which happens due to, right? Actually, that's probably not the best place to put it. Maybe I can't erase that, so I'm going to erase this red one here. The secondary structure is due to complementary base pairing in the stems that generate the stem loop structures. And this even leads to additional folding up on itself. So over here they're showing you, here's the green part of this, here's the yellow part, here's the red, and this whole part is out here. So you get this folding up into a tertiary structure when it folds on itself. What is all of that kind of bonding? It's all going to be non-covalent bonding. Generates this tertiary structure. So the question, once again, you know the structure of DNA, you know the structure of RNA. I said DNA can't be the original self-replicating molecule. Well, get this. RNA, number one, it has the information. It can store information in the bases, and it can actually copy itself. It can act as an enzyme. And the person who found this out also won a Nobel Prize. Excuse me. Sorry. Won a Nobel Prize. This enzyme, because it was made of RNA, was called a ribozyme. The ability of RNA to do both of these things is why we think that the first life form was an RNA that became enclosed in some sort of membrane. And this was the first cell. So how do we know that RNA can act as a catalyst? So Altman and Czech, like I said, won the Nobel Prize for this. Notice what year? Not that long ago, basically 1990. Okay. Science is really making huge strides, and we're in the midst of it. What they did was they looked at a particular microorganism called a tetrahymena, a non-pathogenic, which means it doesn't make you sick, free-living ciliate protozoa. So this is a cell that lives in water, and it's able to catalyze the hydrolysis and condensation of a phosphodiester linkage. What does that mean? What it means is, right, so these guys, Altman and Czech, is that how you spell that? Altman and Czech, were able to demonstrate that RNA, and they could take a sequence of RNA, and who knows how long it needs to be, and use it as an enzyme to add, okay, let's say this is a DNA molecule, and you have a nucleotide here. Alright, here's our base. If this is DNA, you need to have an H and an OH and then a CH2, OP, 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 O. Oh, plan ahead. That's a double bond, that's a single bond. Double bond, single bond. Okay, so if this RNA is the ribozyme, it is able to place this next nucleotide on this sugar phosphate backbone using the energy that's stored in these bonds out here. These bonds are called phosphoanhydride bonds. Very, very, very much energy. Very much. That's very poor English. A lot of energy is stored in that bond. Well, how did they do the experiment? So this is another experiment that I would like you to be able to explain, and let's just walk through it. Alright, at the top you see that in their experimental setup they have some random sequence of RNA. Alright, and they've made these, they've just generated them randomly by putting together a whole bunch of RNA nucleotides. Each one is going to be different than the other. They mix one of these in a tube with what we call a template and a primer. And so the template is this 5' to 3' strand. In this case, you're using RNA. And this is template, and then they put a little primer on here so that it gets started. And they say, can this particular molecule, the big molecule that they generated, can you see the addition of nucleotides to that particular primer? If you do, it means that the original big RNA molecule acted as an enzyme. If you don't see anything, then it didn't act as an enzyme. Well, how do you do this? You do it over multiple different tubes. You make tons and tons and tons of these randomly generated RNA molecules, and in thousands of test tubes you do the reaction. this is what they got when they looked at this. So this is how they analyzed the results. They used radioactive, a P32 molecule of phosphorus, which is radioactive. And so on each nucleotide it had a radioactive tag. And what they did was they looked at that primer and they said, does it get bigger? Do you see that it gets bigger over time? So over time, at the 0 hour, this is how big the primer is. And then at a half hour, you start to see some additional bands, which means that you've added some nucleotides. At 3 hours, oh look, we're even making more and more. And by 24 hours, you see tons of new product. So this would be the test for one test tube, right? This is one test tube with one long RNA acting as a catalyst. You might have a second test tube where you see none of this. Okay, in this one, it would look like nothing, nothing, nothing. Sorry, they should all be exactly the same height. So they took these guys, the ones that they found that could generate this phosphodiester bond, and they did some mutations, and then they looked again to see if they could get even longer strands of RNA made. That was the original experiment that suggests that RNA does contain catalytic activity to reproduce, and we know that it has information, right? A template, a blueprint, in order to make a new strand. And so this is the real data that suggests that RNA, okay, we live in an RNA world, that RNA was the first living organism. Okay, that's going to be it for this part of it. We're going to work on genetic code in class. Come to class knowing what we just did, okay? The genetic code is hard, and we're going to have to do some practice problems in class for that. Okay, see you tomorrow.